I'm Koji YASUTOMO, Professor of the Department of Immunology and Parasitology, School of Medicine of Tokushima University. The focus of our study is the identification of genes or molecular networks that contribute to or suppress chronic inflammation. Inflammation is a necessary physiological process, but when it is too strong or persistent, it precipitates many diseases, including autoimmune disease, allergy, diabetes, and cancer. We have used two approaches to tackle these questions. First, we have tried to identify the causative genes in familial inflammatory disorders. By establishing murine strains that have causative gene mutations, we have addressed the molecular mechanism underlying the disease. With this method, we have discovered 5 novel gene mutations. Presently, we are continuing to search for causative genes in both familial cases and in sporadic cases. This experimental approach depends on the identification of the causative gene by its location on chromosomes, but not on the function of the gene products. This approach identifies the causative genes beyond the roles expected from known gene product function. One example is the discovery of a mutation in PSMB8 in autoinflammatory syndrome. PSMB8 encodes beta-5I, a subunit of immunoproteasomes. Before our discovery, there had been many studies of immunoproteasomes, but the functions of immunoproteasomes in humans were not clear. The identification of the mutation in PSMB8 proved the function of immunoproteasomes as suppressors of inflammatory responses. In addition, immunoproteasomes were originally identified in Tokushima University in 1994, and human patients with this mutation in immunoproteasomes were also first identified in Tokushima. This is a very interesting and surprising coincidence. The second approach of our laboratory is to evaluate the molecular regulation of T cells, particularly memory T cells, because the persistence of pathogenic memory autoimmune T cells lead to irreversible autoimmune pathology in tissues. We have found that notch signaling is essential for the maintenance of memory CD4 T cells by regulating glucose homeostasis. Now we are evaluating molecular networks in mice as well as human memory T cells with multi-omics approaches. We think this study will be important for determining the underlying mechanism how T cells choose their cell fate toward memory T cell. Also, this study will be essential for developing novel vaccine against pathogens and cancer cells.